Hi, everyone. I am Cristina Pertil. I am the CEO of Plio Surgical. Plio Surgical is a spin-out from Trinity College Dublin, aiming to disrupt GI minimally invasive surgery. Our team is addressing the most severe complication of GI surgery, a leak that can occur after the surgeon removes a diseased portion and reconnects the healthy ends, which is called an anastomosis. When this happens, the patient can become very sick. They may develop sepsis and require multiple reoperations. This adds more than 12 days to their hospital stay, and more importantly, it may delay future treatment. On average, the leak rate is 9%, but it can be as high as 20% in certain procedures. This represents a huge burden for healthcare systems around the world. Despite this big problem, the current standard of care hasn't changed much over the last 40 years. Surgeons mainly use surgical staplers to perform an anastomosis. They are telling us that innovation is required to improve outcomes for patients. Also, with the increase of minimally invasive procedures, surgeons tell us that conducting an anastomosis inside the body, so intracorporeally, is very challenging. So less than 10% attempted at the moment. So this is where Plio comes in. It provides a novel and clever approach to a GI anastomosis. The Plio device consists of 2 rings that can be compressed and delivered in a fully laparoscopic procedure. They have specially designed tissue anchors that allow them to stay inside the lumen without the need for stapling or suturing. The rings are aligned with magnetic elements that bring them together and compress the tissue between the rings, allowing the healing to happen in less than 30 days. After the healing occurs, the rings detach and pass naturally, leaving nothing behind in the patient. So what makes Plio unique in comparison to the competition? We are the only company that has patented tissue anchors and the elastomeric material that allow the rings to be anchored inside the lumen without any stapling or suturing. This makes it easier to perform a minimally invasive delivery. Also, it offers a wide range of applications. For example, the rings can be made in a range of sizes to suit the anatomy where they are placed. The technology was validated in extensive preclinical studies with excellent results. We conducted the testing in IHU Strasbourg and achieved a very good anastomosis and good healing. The device passed within 10 days, and we obtained excellent histology results at the study termination of 29 days. From the start, we have worked closely with surgeons to develop the Plio device. We received very positive feedback and support from prestigious institutions such as the Mayo Clinic in the US, with which we are now discussing a collaboration agreement. Surgeons are telling us that they see a wide range of applications for our device. The beachhead application will be in colon cancer, but the Plio device can also be used for other procedures such as the Crohn's disease or bariatric weight loss surgery. This represents a substantial market potential of more than 3 billion euro. Our route to market is well defined. We will target a market entry in the US first. The Plio device will be classified as a Class 2 device and follow a de novo pathway. We plan to take advantage of existing reimbursement codes and employ a small direct sales team targeting key hospitals where we have established relationships. Following this, we will expand to the UK and European markets after securing the necessary clearances. We have a team with a strong medtech innovation track record that can execute this strategy. I bring experience from my time with GE Healthcare in the States, where I develop patient monitoring products. Professor Bruce Murphy has successfully spun out technologies from Trinity College before. We are supported by a network of business and clinical advisors with relevant experience in the field. Our efforts have not gone unnoticed. We are also recognized by admission to the very selective MedTech Innovator Accelerator in the US, which provided us with access to partners such as Johnson & Johnson. Looking ahead, we are raising a 3.6 million seed round to conduct a first-in-human study and progress through regulatory approval. We hope you can join us on this journey to establish a new standard of care for GI anastomosis and improve the lives of millions of patients around the world. Thank you.
Are you addressing both intraoperative leaks and postoperative leaks, or only postoperative leaks?
Well, the majority of the leaks actually occur after the operation. So that is, they're actually really hard to detect, and often the patient becomes unwell about day 5 after the operation. So during the intraoperatively, the surgeon can perform certain tests to check that there is no leak, but afterwards is the main issue that they don't really know which anastomosis is going to leak and which one is not.
Yeah, well, depending on the data you look at, I mean, some studies suggest that it's about 50-50, but so I understand you're only addressing post-operative leaks, right?
So 50-50 is that probably after the patient has been discharged? Is that maybe I didn't understand the question. So 50-50, I think 50% occur while they're still admitted in a hospital in the 7 days, and 50% after they were discharged.
Well, just so I get a very simple understanding, is the technology addressing post-operative leaks only or both? So my understanding is it's only post-operative leaks, right?
So what the technology does is really offer an easier way to conduct the anastomosis. So we're offering an alternative way for surgeons to do the anastomosis without the use of stapling or suturing. So we think we provide a better seal and a better healing of the anastomosis. So if you look at histology results of magnetic anastomosis like ourselves versus stapled, the healing is superior for magnetic anastomosis.
Okay. And just one last question. Can the device, does the device inform the physician real time whether there is a leak or not? Or you're just sort of counting on the clinical evidence you're going to prove in the clinical trial to say, okay, well, by using this device, there's not going to be any kind of leaks. And the underlying question is, unless you can provide some real-time data, physicians might be tempted to at least for some time to still conduct some air bubble tests.
Yeah. So no. Well, I guess the question about whether you can detect leaks would so it would require sensor on the device. So we were not planning to have that in the first generation. Maybe we were considering that because we know it is an unmet need for future generations, but not for the device that we are developing right now that we're planning to bring to the market. So that, but what would be interesting, for example, because the device passes, it doesn't stay there. It does provide, I guess, verification that the healing has occurred. And so, for example, a lot of patients receive a stoma because the surgeons are not sure if they're going to leak or not. So what the surgeons have told us that if they know the device passed within 10 days, they probably could reverse the stoma sooner.
Thank you.
So there were some experiments with rings prior to your product, and I believe there was a problem with the uneven pressure between those rings, why they from one side to another when applied and when further on the healing is in the process. So how do you apply pressure between those two rings?
Yeah, so the pressure is applied by the magnets. So we have done quite extensive research and literature on what is the magnetic force required to apply the force for the different sizes of the GI tract. So as you probably know, the colon or the small intestine, they have different size of the intestinal wall. So we have a certain range that our device is going to apply, the magnetic force. We've tested that.
And how does that mechanism then differ from the current stapling method by applying those two segments to each other?
Yeah, so what the difference is that, you know, what we do is we actually bring I don't know if you can see that. For some reason I cannot see myself on the camera, but so you can see these two rings. They have this special tissue anchors. So the tissue is actually gripped by this special anchor. So they literally just go together, and the magnets compress the tissue between them. Whereas with the stapling, there isn't that compression force. They just, you know, they're stuck together by the staples, by the metallic staples. So they're always kept together. Whereas ours, you know, compresses the tissue. So the tissue between the rings, because it is compressed, necroses and dies off, which allows the rings to pass. And so there's nothing left behind, whereas the staples always stay in the body, you know, so you leave behind something like 120 staples or so.
Thank you.